Hi, I'm Sam.
I'm Nia.
I'm Emily.
And this is Chapter 3.
Cells.
The living units.
This is the fluid mosaic model of the membrane structure. This is the phospholipid bilayer. It makes up the membrane with hydrophilic heads and hydrophobic tails. The hydrophobic tails are scared of water, and the hydrophilic heads love water. Glycolipids are cell surface markers, and they help stick together. Cholesterol keeps and helps stabilize the membrane. Membrane proteins help transport material in and out of the cell. Integral proteins build into the structure, and they are transmembrane proteins. Peripheral proteins are inside or outside of the cell, of the edge of the plasma membrane. Functions of membrane proteins. A. Transport. These can be channels or carriers. Channels are tubes that things can go in and out of. Carriers are the passageway. B. Enzymes. These catalyze. C. Receptors. They pick up a message from the outside of the cell, send a message into the inside of the cell, and these are mostly for molecules that can't get in. D. Intercellular adhesion. This joins cells together. E. Cell-to-cell recognition. This is how cells recognize each other. F. Attachment to cytoskeleton and extracellular matrix.
Microvilli increases the surface area of the cell, making more room to absorb things.
Membrane junctions. Cell membranes come joined together. There's 3 different types. Tight junctions are waterproof connections between cells. This is where you don't want any leaking, especially in the stomach, esophagus, and the bladder. Anchoring junctions, also called desmosomes, are really strong connections between cells. Gap junctions are channels between cells, and this allows communication between cells, especially in cardiac muscle heart cells. Movement of materials across the plasma membrane. Selective permeability of the membrane lets some things through but not others. Water can freely move through, but proteins cannot freely move in. Passive processes don't require energy. Simple diffusion moves from a high concentration to a low concentration. Facilitated diffusion is when you have channels or carriers, and this requires no energy.
Osmosis is the diffusion of water across a selectively permeable membrane. Hydrostatic and osmotic pressure is caused by pushing on something, and it comes because of contraction of dissolved solutes.
Tonicity is the ability of a solution to change of the cell by altering the cell's internal water volume. There are 3 types. Isotonic, the concentration of either side of the semi-permeable membrane. Cells remain their normal size. There is no gain or loss of water. Hypertonic, the water moves outside of the cell and the cell will shrink. Hypotonic, there is less salt and more water outside of the cell, so it will move into the cell. The cell becomes bloated and will burst. Active processes require energy. Active transport is the sodium-potassium pump. 3 sodiums can go in and 2 sodiums can leave. Vesicular transport has different parts. Exocytosis is bringing material out of the cell. Endocytosis is bringing material into the cell. Phagocytosis is the cell eating things like large particles. Penocytosis is the cell drinking liquids. Receptor-mediated endocytosis is specific molecules put receptors outside of the cell when they're full. Then things are brought in little by little.
This is the cytosol, all of the stuff inside, the clear part. This is the mitochondria, it looks like kind of like carrots, and it produces the ATP. These are free ribosomes. Ribosomes are what makes the proteins. These are also free ribosomes. This is the smooth ER, which is endoplasmic reticulum. This is the rough endoplasmic reticulum. And it's what the ribosomes are on it. They're membrane bound. This is the Golgi Apparatus. It packages, modifies, and transports cellular products. This is the liposomes. This is the perosomes. This is what contains the peroxidase.
Some things that are not on the model are the centrosome and centrioles, and that makes the mitotic spindle, and they're made of microtubules, and cilia and flagella, and they make cells move. They're usually outside of the cell. Inclusions of the cytoplasm include glycogen, lipid, and melanin. Glycogen is a polysaccharide that is the chief carbohydrate and is used for storage material. Lipids are various substances that are absolute in nonpolar organic solvents. And melanin is the pigment given to us. It's produced by cells called melanocytes. That is the nuclear envelope. That's a double-layered membrane enclosing the nucleus of a eukaryotic cell. Nucleoplasm is the substance in the cell nucleus. In this cell, there are two nucleoli, and there are small, dense, spherical structures in the nucleus of a cell during interphase. And chromatin is the material in which the chromosomes of organisms other than bacteria are composed, and that is an active form of DNA.
This is interphase, where the cell is being a cell. This is prophase, the first stage of cell division. The chromosomes become visible as paired chromatins in the nuclear envelope. This is metaphase, second stage, and the chromosomes become attached to spindle fibers. This is anaphase. Chromosomes move away from one another to opposite poles. This is telophase, final stage, sister chromatid reach opposite poles. This is cytokinesis, the cytoplasmic division of a cell at the end of mitosis and meiosis, bringing about the separation into two daughter cells.
The roles of RNA. RNA is what makes the proteins to order. mRNA is the messenger RNA, gives you the primary structure of RNA. rRNA is the ribosome RNA, stuff that makes the ribosomes. Ribosomes are the machines that put the proteins together. tRNA is the transfer RNA. It brings the appropriate amino acids.
Copy from the DNA. The DNA stays in the nucleus. Translation is making a protein out in the cytoplasm.
There goes the cell.